Good afternoon, everybody. My name is John Field. I'm the Executive Secretary here at the Pacific Salmon Commission. I want to welcome you to the latest installment of a seminar series put on by the Commission over the last year or so with monthly installments. Today will be the 8th in a series of these virtual seminars. They're intended to provide information on environmental change, its effects on salmon across their life cycle, and salmon management from both Western and Indigenous perspectives. The series, just like today, the series in the past has featured noted speakers from the US and Canada, and then we'll do a facilitated Q&A session after each speaker. Today's seminar is being recorded. The final recordings will be put up for public consumption, and for those of you who missed ones in the past, you can go to the PSC website, which is psc.org, or to our YouTube channel. If you just search YouTube for the Pacific Salmon Commission, you'll come across all the today's recordings and all the past recordings as well. Today's event is entitled Environmental Variability and Hatchery Effectiveness. We have two great speakers joining us today, Dr. Brian Riddell from the Pacific Salmon Foundation, and Dr. Brian Beckman from NOAA Fisheries in the United States. I'll introduce Dr. Riddell first. He'll be the first of our two speakers. I'll stop sharing my screen at that point and let him present what he needs to. Brian is currently the science advisor at the Pacific Salmon Foundation after serving as president and chief executive officer for a decade there. Prior to joining the Pacific Salmon Foundation, Brian worked for 30 years in salmon research and management at the Department of Fisheries and Oceans in Canada, where he co-authored the country's policy for conservation of wild Pacific salmon in 2005, and then provided science advice to the Pacific Salmon Treaty and the negotiations that happened throughout his tenure there at DFO. At the PSF, the Pacific Salmon Foundation, he established the Salish Sea Marine Survival Project and the research network that was associated with that. Of course, Brian, those of you who know him know he has won many awards and honors for his work, and I'm proud to call him a colleague with his time here at the Pacific Salmon Commission. With that, Brian Riddell, I will turn it over to you and stop sharing my screen.
Thank you, John, and good to see you. Since I'm no longer involved with the commission, so I'm going to share. Okay, well, thank you very much, John. Overly pleasant introduction, and no longer a science advisor. I'm sort of a contract hanger-oner, and that, so stepping into retirement slowly. I think you'll see today's talk from the Salmon Foundation is very, very consistent with the theme of environmental variation and hatchery effectiveness. I was to have a co-author with me today, Isabel Pearsall, who's now the director for the marine programs at PSF. And with our snow over here, she has no power, and so she will not be able to participate. I guess the other part of this is kind of a bit of a get back, because Michael Folks assists us on one of our advisory programs. And when this topic came up, I really couldn't say no to him when he asked. So what I'm going to do today is give you what will have to be a very quick overview of a large program funded by the BC Salmon Restoration and Innovation Fund. And full credit to the PSC, this project is a direct development from the Salish Sea Marine Survival Program, and the Salmon Commission really got us going in 2013 there. With a $5 million contribution to the program to Long Live the Kings and PSF. And after we received that, we were able to get the rest of the money. In the end, that grew to a total commitment of $23 million invested for salmon conservation, rebuilding, science in the Salish Sea and the two countries. The connection with the hatchery effectiveness really follows from our in-depth study using pit tags at Cowichan River on the Chinook hatchery and wild production. And a very consistent outcome of that research is that the hatchery fish that were tagged in the wild in 4 different sites and paired samples with wild and hatchery had a pretty consistent 1/3 survival of the natural fish. And we invested heavily in the collagen there. We will now have put on over 100,000 tags, pit tags. And in the study period 2014 to 17, we applied 90,000 pit tags. This was the first really major application of pit tag technologies in Canada, and we've built a large receiver array across the Cowichan to get good recovery. And in all 4 locations where we tagged Chinook salmon, so we had the Ind River, along the beach in the very near shore, and then purse seining in Cowichan Bay, and then micro trolling just slightly offshore. In all 4 situations, the wild fish came back at much higher rates than the wild. This has actually led to another major study called the bottleneck study, but that will have to be discussed at a separate time. So I'm just going to... So what we did following the Salish Sea Marine Survival Program, when the BC Shrift funds became available in 2018, because of the long-standing relationship of the Salmon Foundation with community programs, community hatcheries, community restoration programs, we saw a real opportunity and a need to do a critical evaluation of how effective hatchery production really is in British Columbia. It's probably not a secret that there have not been in-depth assessments of our salmon enhancement program for over 20 years. And there had been some early criticism in the early 1990s, but it's been pretty quiet in there, and a lot of work is going on. So we thought it was certainly time to really do a very serious review of this program. It is without doubt the largest single program annually in the Pacific region of DFO. So this is just introducing you to our team. It's a large team. You won't know all these names. Some of them are young people that we brought in. You will know some of the people on the advisory panels. So the independent science panel was really overview or give us advice from other such reviews done in the United States, for example. That was provided by Craig Busick, Mike Ford. And then the province was Tim Isaki, David Suzuki Foundation, Jeffrey Young, and Dave Willis is with Salmon Enhancement Program. Later in my talk, we'll go into the analytical type of work. These are without question the most complicated mathematical models we've developed to look at enhancement in BC. And a lot of the advice were provided by Dr. John Moore, SFU, Michael Folks, DFO, Sean Cox at SFU, and Carl Walters at UBC. Okay, so these are the components of the full program. I think you'll see immediately it's very ambitious. When Michael asked me to talk about this, you're going back a few months here, and I thought we would be finished. I have to apologize, we are not. We have finished some significant portion of it, and we're very near complete on other portions, and some we're still doing, really testing our analyses, to be honest. We did receive information just today that the first scientific publication has been accepted by CJFAS in that, and I will give you a very, very brief overview of that. So there are 8 components. I'm not going to talk about the spawning channels today because we simply don't have it well organized in that sense. That's being done by Cam West, who has a long history in the Salmon Enhancement Program. You can see reports for some of these on a website. If you go to marinescience.ca, that'll bring you up within the PSF website, and you'll see about 10 folders there with various names where you can see publications in Isabel's program. I'm going to touch very briefly on the first 7 of these projects here. So the very first one is really building from our research we've done with Christy Miller-Saunders, and that's part of the Strategic Salmon Health Initiative. What we wanted to look at here is really look at the opportunities for new studies in hatchery environments using genomics. The report that was first written by Amy Lee Howd is called the Omics report because there's various types. of genetic analyses that can be done. I've only taken out one example here for you related to our parental base tagging and the SNP genotyping that we use in the PSF. So Amy did a very nice review of opportunities for us in all the various types of modeling and analyses that can be done. But then we wanted to really apply it to a hatchery environment, and we asked Wendy Vandersteen to do a report following from Amy's about how you could apply this to learn within the hatchery environment. Both of those reports are complete, and they are on that website. The second step for us seemed to be that we wanted to bring up to date the literature reviews that people have done in the past about Pacific salmon in hatcheries. And we wanted to really focus on the literature associated with interactions between hatchery fish and wild fish. When we did the initial screening, we ended up with a huge array of salmon hatchery type literature, almost 5,000 of them. But when we really focused down into some that had information content related directly to, excuse me, interactions, we got down to only 85 publications here. And they were grouped into these categories I have: fish health, competition, outcomes. This is really looking at the productivity changes, return at age, size at age, and survival rates of the wild fish. As this slide presents, it's pretty clear that the majority of these published papers and reviews, there were a few reviews involved, really indicate negative effects of interactions at this time. The next is that there was neutral or no obvious effect, no demonstrated effect, and only 2 of 85 really showed a positive benefit of growth of the population or productivity, for example. Right, so not particularly different from the past, but these 85 are focused only on the interaction between hatchery and wild fish. This is complete. The final edits are being done. The author had used a lot of statements about the percent of the differences and so on, but sometimes it was the percent. 67% is 2 out of 3 studies. So we thought that was a little disingenuous to really express it that way. So we've included all of the direct comparisons by number. So you have the percentage and you have the actual number of studies involved. The first really big review is of the community-based hatchery program. Now, for some of you that don't know the Salmon Enhancement Program as well as others here, it's a very complex, large program. There are 23 what are called major facilities. These are under the operational management of SCP. That is not what this community hatchery program is about. So the community hatchery programs are smaller scale, widely diversified, and they are actually assisted by 15 professional biologists in BC we call community advisors. The study that we did had to take a subsample of all of the community programs. We ended up actually doing 32 reviews of facilities, and that's actually a pretty good sample overall. If you look at all of the projects included in the in the community economic development, so these are a little bit larger scale. They would be programs like the Cowichan. Or if you looked at the total production of releases from these 32, it would be 82% of the total releases. So the one that we didn't really involve tended to be smaller scale of the community programs. You can actually look at a lot of the information that are going to be in our reviews in the integrated fisheries management plans that DFO publishes each year. And each year in there is a Excel file basically of the planned releases and the actual releases from all facilities in British Columbia. So when we completed all of these interviews, what we really found here is that there's extremely good consistency between the planned releases agreed to by SEP and the communities. But what was missing is that a Salmon Enhancement Program publishes what they call best management practices, and they are what are to determine how community hatcheries operate. There were a number of serious inconsistencies there, not a large number of them, but some serious differences that we noted. Not too surprising, almost 3/4 of all the facilities interviewed indicated that they needed more money, and that was one of the most serious deficiencies that we identified. The biggest concern to me is many of the best management practices stipulate that you should be using hatchery fish for that and wild fish for something else. But in reality, the minority of the projects actually do sufficient marking to really apply best management practices. So there's definitely a breakdown in the assessment role there, or evaluation side. And bottom line is, much of the benefit from the community-based level comes from these larger CEDP facilities. Many of these are managed by First Nation communities, and there's the smaller public involvement programs are very diversified, but involve much smaller numbers of fish. There's also an important educational component, but that component wasn't included in this review. They deal with very small numbers of fish, but they're widely diversified. The recommendations that we've included, just very briefly, we talk about the increased support, improved communication to ensure some of the best management practices are actually applied. There is a need for updating the exchange of data. And the other part, excuse me, we really have to do a better job to evaluate community-based projects. It's very difficult. We can do the interviews, but we really couldn't come up with any quantitative evaluation based on the absence of tags. The fourth level is the first one, and this is what a paper has been received and just accepted today formally. This was a large analysis of all of the data for Chinook and coho from 1972 to 2017 brood years. And this is because the analysis looks at the releases in a year and all of the subsequent recoveries. Plus, as we started to build these models out, we involved a number of covariates, ocean temperatures, PDO, and then the predators and year effects. The outcome of this is complicated models, hierarchical models, that really shows that we have declining survivals over time, but it's against a large background of environmental variation, just as the seminar series is addressing. I'm going to show you only two examples. So this is the plot for Chinook and coho of all of the releases and recoveries, the top for Chinook and the bottom for survival. It looks like a bit of a mess at first glance, but the lines would give you more information. This, you really have to follow through the report. Bottom line to me is there's really not a lot of talk about optimality in any of this. There is a huge amount of environmental variation, and you have to really consider that within the next figure. This is the trend for year effects through that entire time series. So both for Chinook and for coho, we have a rather alarming decline in the survivorship from these hatchery releases. You can see the variation against the trend line. It's not that significant over time. A lot more noise early on, a lot less noise as you go further on, but a very strong negative trend across our enhancement programs. And these would be essentially from all of the major and operational programs and some of the larger CEDPs. Now, the other element that's important to us is the biological characteristics of the stocks over time. This turned out to be a huge data collation and validation problem. And we knew the problems that we wanted, or I'm sorry, the parameters that we wanted to look at: size at age, age of return and maturity, and that. But we weren't able to do that without spending a lot of time working with some groups in DFO to organize what data we had. So the data that we actually got to use, we ended up having information we could look at for 75 different BC Chinook stocks. And they came from a variety of different databases, some with the enhancement program. The others are what we call the bio data from the stock assessment program, some specific hatchery that had unique programs, Kitimat, Nechako in the Upper Fraser, And then a lot of the information we could also take from quota-wire tags. And this is the same database that Oldberger published about a few years ago. So that's just where the data is from. This is the sort of couple of summary slides for you here. I've got two slides that basically show you the same thing here. We're talking about enhanced return, and this is the change in age composition. Across the horizontal axis is the change in percent per year. So you can see a pretty significant decline, for example, in Quesnel age 2 Chinook, about a 20% decline per year. That seems very, very high in that. But at the upper thing, Shuswap at the bottom, you can see that you've got an increasing number of ocean age 2 fish coming back. And then you can see the decline in age 3s and 4s. But if you just look at a few of the outcomes here, this is from one database, but here's the exact same parameter from a second database, and they're not the same. So this is the problem we have in collating this data. Excuse me, one of the things we really have to do is validate some of these and find out if we're actually comparing an apple with an apple amongst these data sets. So that's why I say some of these things that we're discovering we want to sort out, but we haven't completed that yet. One that I think is certainly of real interest to us, if we look at just size at age, across the board, we are definitely seeing declining size at age in BC Chinook. There might be some variation in this between data sets and all the rest, but I think most of us would agree that these are some pretty substantial changes that are notable and would certainly have an effect on the productivity of these populations. So an issue of concern here are key messages that we had a really difficult time sorting out a lot of this data. We weren't even able to undertake the coho or chum assessments, and we didn't even try at sockeye. The EPAT and the biodata are useful because they're large samples over a number of years and that pretty consistently collected, but we have trouble in comparing some of the data sets. But the bottom line to me is I don't think there's any real question. We are seeing some concerns for decreasing size at age and sex and decreasing age at maturity in BC Chinook. So this is obviously a point of continued concern. Now, the more detailed analyses that we want to look at in terms of hatchery effectiveness, we've looked at this in two ways, really. One is we want to look at whether or not we see changes in productivity changes within natural populations, and then whether the hatchery fish have effects amongst natural populations over spatial and temporal scales. So we've had to actually develop means to actually do these analyses like this. So what I'm going to show you here is just a very brief overview of what our analysis is. So we are looking at enhanced contribution of Chinook, coho, and chum to harvest. That, where are they caught? We're probably well aware of that more than others. And are some hatcheries more effective than others at producing catch? We'll all be aware of the data needs. This is just one of many, many plots that you can generate, as we all on this phone will realize. So what we're looking at here is the interior South Coast coho, and the plot that I'm showing you here just is one way of looking at the efficiency of production from different hatcheries. What we find is you've got the box plots where you've got a mean level of production and the variance and then the range. All right, so there are about 4 different ways of presenting this in our software analyses that will be available. So we can look at the total catch, CWT distribution, enhanced contributions, and efficiency that we look at. Now, this is near completion, but not final. So the type of thing that we have looked at is that we want to go on and look now at not just harvest, but rebuilding of natural populations. So the questions that we agreed to look at through the consultation with SEP, does enhancement increase the total and the natural origin spawners in natural population? What happens when the hatchery production stops? And you can look at the efficiency aspect again, and then whether or not these outcomes differ by regions and species. As we probably all know in this meeting, it's a demanding task to do all this because you really have to use all the data available to you. and that has limited some of our analyses. But the way we're looking at this, every single system in the area that we look at, we have what we call a dashboard by stream. So the picture really on the right screen here, you can look at the escapement trend. This is total return. In this case, this is the Sarita River on the west coast of Vancouver Island for Chinook. You can look at the releases and the related hatcheries directly into the Sarita. Then you can look at the escapement returns. And as the hatcheries come in, of course, you have natural origin and total and wild fish producing. And then at the bottom line, we have taken on some of the parameters that developed in the US studies by looking at the proportion of hatchery and spawners and the proportion natural, etc. And this will be available for every population in an area that you study. So this is the dashboard that you can see. And I'm just going to show you one example here. We've also got a generational presentation where you can look at the returns over time, and the color scale here is really just about the generations. And then later on, you can look at the natural origin spawners. So these are all the fish spawning in the wild over time. And then this is really looking at the contribution or the wild spawners remaining that would have the proportion of natural spawners here. It's not a positive story in many cases, and the rebuilding effect is really something that we're starting to look at very carefully. So the bottom line to us is we definitely see an increase in total abundance when the hatchery fish are returning. You get an increase in the total number of spawners. Natural origin spawners, though, do not sustain themselves in many, many cases after you stop enhancement. So this is a serious concern to us in terms of how effective rebuilding of the natural spawners really are. And that there's no question that the hatcheries produce fish effectively, but the ongoing effect of rebuilding is certainly less evident. So we wanted to then look at the interactions over larger spatial areas, and this is the sort of second analysis. I just went through this takeaway, so given the time, I won't repeat it. So the second question is, are the hatcheries influencing wild populations? So we wanted to first off look at the productivity of the natural populations through time and space. And then we wanted to see whether or not in the face of hatchery production or in just spatial coherence amongst natural populations, what we could say by looking at groups of streams within conservation units. We've done this because there have been a number of reviews and a number of efforts to standardize data within some of these. So this is the data source. The Pacific Salmon Explorer, of course, is the software developed by the Pacific Salmon Foundation in collaboration with DFO. Nelson is the work out of the United States and Puget Sound. He looked at 8 units of Chinook stocks. Winther, this is the Kitsum-Kalem population in the Skeena, and the inner South Coast chum. And chum are interesting. There's actually some good data sets, Area 6, Area 8 in the north, and then the southern inside chum. So those are the ones that we chose to use because they have had greater scrutiny in terms of data quality as we do this. And we looked at really 2 steps. One is within single conservation units, looking at Ricker stock recruitment curves, just the standard ones and time varying, looking at the correlation and productivity amongst different populations, and then the spatial work I referred to. And now some of the work is being done by Landmark Consulting, and they're doing much more complicated multi-conservation units and much more complicated Ricker stock recruitment models here. All of this is very near completion, but really we're getting just preliminary results, so we're not comfortable and really widely sharing it yet. It will be very soon. Now, to look at the spatial effects, one thing that we really wanted to look at in detail is how relationships from a point source like a major hatchery, such as the Kitimat in Area 6 in this one example, But how do you actually set up the data to look at the spatial effects over time in complex areas where the animals could certainly migrate into different locations? So we've actually spent quite a bit of time, and Andy Rosenberger is probably the best person to talk to about this if you want. This is just an example about how we're measuring distance between the point source of a hatchery and the spawning grounds for the natural populations. Right? And the outcome of that then are a number of different types of spatial analyses. And one of the things that we've discovered is that depending on the metric you use over time, you get slightly different outcomes. So what I wanted to show you here is just one single example. Excuse me. So the top, or all four of these are different types of metrics about trends in the spawning escapement for natural population and from a certain distance to a hatchery. I think it's interesting. One of the things we had not anticipated is you get different results with different metrics. And one of the most interesting plots is the bottom right, where this is a simple generational moving average. And you certainly see much more sort of strong geographic clustering there in populations, some negative and some positive in the scales that we have. So this is the work that we're really trying to understand now and we'll be working on in the near future. And I think that's it. I'm sorry, it's definitely a large scale study. I will provide a PDF of all of this. And as I say, we will be finishing these publications over the next few months. So I'll leave it at that, John. Thank you.
Thanks very much, Brian. I appreciate that. And I'm sure you'll get questions as we move on here. Our next speaker is Dr. Brian Beckman. Brian is a research biologist with the National Marine Fisheries Service at the Northwest Fisheries Science Center in Seattle. He is also currently serving on the Committee for Scientific Cooperation, or the CSC, within the Pacific Salmon Commission. Brian is a physiologist with a focus on assessing hatchery salmon performance and designing hatchery rearing strategies so that release smolts meet performance goals. In addition, he has a separate research interest in how marine environmental conditions influence growth and survival of juvenile salmon in the Northeast Pacific. And this interest is responsible for his participation in ship-based research surveys from the Oregon coast to the Bering Sea. His talk today represents a fusion of these separate research areas, and the title of his talk today is Marine Environmental Variability and Salmon Hatchery Rearing Strategies, Match or Mismatch. With that, Brian, I will turn it over to you.
Thanks much, John. Thanks for the introduction. And thanks to Brian for a really comprehensive talk about BC hatcheries. And I'm still engaging with that and trying to move in my own talk. And I will. So I'm Brian, I'm from Seattle. And let's go. So this first slide is basically a quick synopsis of my career. For several decades, I've been working in hatcheries on different marine regimes and how they influence subsequent survival of fish. Subsequently, I started working out in the ocean and working on how varying ocean environments affect salmon survival. And so this talk represents kind of a smashing together of those two research themes. And I'm going to end up focusing on one experiment at the end that I'm pretty excited about. So here's an outline. I'm going to talk some about hatcheries. This is fairly opinionated on purpose, and it's about Chinook, and it's primarily about the Columbia River. I'll talk a little bit about the Pacific Ocean. Briefly, if you've been following this seminar series, it's something, or it'll be nothing new to you. And then finally, hatcheries meet the Pacific Ocean, and particularly in terms of variability. So this slide represents really my conceptual view of Chinook salmon, which is the life history is quite variable within populations, between stocks, and particularly the size, age, and season of ocean entry of juvenile Chinook salmon varies. So hatcheries are different. This is a shot of two books that really helped me understand the development of hatchery programs in the Pacific Northwest. And hatcheries really initially were based upon this idea of industrial agriculture. So industrial agriculture, if you're bringing chickens or pigs or corn or whatever, is really based on high production. It's based on efficiency or the cost per unit of production. And this efficiency is developed through standardization. So variation is eliminated. In hatcheries, releases fish are the same size and the same age, and they're released at the same time. And really that goal is based upon the most smolts for the fewest dollars. So if we go back to the cartoon, all the variation is gone. There's single lines and all the fish at the same time, the same size. And that's the hatchery's goal. That's what they wanted to do. So there are some assumptions underlying industrial smolt production. These include unlimited ocean carrying capacity, positive relationships between smolt release and adult return, uniform marine environments across years, and all smolts have the same performance. So we'll look at these assumptions as we go through the talk. So let's look at production strategies in the Puget Sound. So this is based upon a paper by Ben Nelson. So basically it's the results of his paper. On the bottom right panel, you see hatchery production in the Puget Sound and the Strait of Georgia. You can see hatchery production ramping up in the 50s, and really peaking in the 70s and declining some post 70s. And so remember this ramping up period in the 50s, in the 70s for later on. And this is a plot of the release characteristics of Chinook salmon from those hatchery releases. Again, the 50s through about 2010. You can see in the 50s, release date was fairly variable, and it's compressed to a fairly common release time in late May. You can also see originally release size was quite variable, and that's also compressed down to a fairly common size, about 80 millimeters. So hatchery production strategies in the Puget Sound have become more uniform and standardized with a common release date and a common release size. So we think about this, and we think about the hatchery release being a bet. So in the Puget Sound, I'll call this the big bet. All fish are going out on the same date at the same size. And the assumption is the ocean's going to be productive and support fish on this date across years. So the Pacific Ocean, I tend to use the PDO, or Pacific Decadal Oscillation, as an index of ocean conditions. There's a bunch of words there at the top that tells you what the PDO is. For my purposes, basically we can talk about the cool phase and the warm phase. The cool phase represents relatively cooler ocean temperatures off the Oregon, Washington, California coasts, and that's good salmon production. In contrast, up in Alaska, the warm phase produces better salmon returns. But again, for my purposes, cool phase is good for the Pacific Northwest. Okay, so hatchery production strategies really developed during the 50s and 70s. And you can see in this green box, that period was really relatively stable, good ocean conditions off the Oregon, Washington coast. So hatchery strategies were developed during a long period of good ocean conditions. So the big bet worked well during the period of hatchery development because ocean conditions were favorable and relatively stable across years. Okay, in the late 70s, code-wire tags really became widespread and applied to many hatchery programs, and hatchery returns could be evaluated. So I'm going to show you some numbers from a number of hatcheries along the Oregon, Washington coast and the Columbia River from 77 to 14. Okay, really simply, this plot shows a correlation of the PDO to standardized returns from these hatcheries. And you can see there's a negative correlation with better returns during cool phase PDO and worse returns during warm phase PDO. So ocean buried and salmon production buried over this period. So this tells us something about the assumptions of industrial smolt production. You know, there's not unlimited ocean carrying capacity. In some years, even though you put out a lot of smolts, you don't get a lot of adults back. And the ocean environment varies across years. So some of the basis for industrial smolt production is perhaps not true. So is the big bet industrial smolt production really the only thing we can do, or there are options? This is totally arm wavy, but are there alternative production strategies that might produce different magnitudes of variation? On the left is basically a squiggle, which shows peaks and valleys, large ones. On the right side is that same squiggle is compressed. So what happens is on the left side, the peaks are higher, but on the right side, the valleys are higher, or the valleys aren't as deep. So what could we do to perhaps reduce variance and think about that as a hatch production goal rather than maximizing returns in every year? So that's what I just said. What are the hatchery's goals? Is it maximizing return every year? Or maybe you want to minimize variance between years? Okay, quick swing through climate change. This plot shows ocean heat. So that's temperature by volume. And you can see there's a steady increase in ocean heat over past 40 years, basically. So this actually is not temperature, but it's basically heat, and it's increasing. And that's simplistically how I tend to think about climate change. Another part of climate change is variation. And this plot shows ocean heat waves, which are basically a surface temperature manifestation. This plot goes from the 80s through to quite recent. And what you can see is the occurrence and magnitude of heat waves has been much, much higher in the last 10 to 15 years. And this is forecast to continue in many models. So we can expect an increasing frequency and magnitude of environmental fluctuation. So this is directly cribbed from a talk by Nick Bond, who's a state climatologist for the state of Washington. So long-term changes in the environment will be masked by increasing short-term variability. So it's going to be hard to see the long-term trend because things are going up and down so fast and so far. And as you create a talk, you kind of develop throwaway lines. And on the bottom, this is a throwaway line, suggesting the average will become more conceptual than real. And so what that, I think, means is the average represents the midpoint between maximum and minimum. But if fluctuations is basically what we see through time, we'll never see average conditions. So as we think about managing hatcheries in salmon, the average might not be all that useful. And that's a thought that I'm still developing, and I'd love to get some feedback on. Okay, so hatcheries meet the Pacific Ocean. I'm going to talk about one experiment conducted in the Umatilla River, which is in Northeast Oregon, in which smolt size and smolt conditions varied at release over a number of years. Why I think you as a PSC audience might care is shown here. The red dots on the left panel show for fish from this hatchery recovered from the 2012 release. And you can see that these fish contributed fisheries from Southeast Alaska through Canada, Oregon, Washington coast, and then most of the fish were actually recovered in the Columbia River system. So this is the experiment. Fish were fed different amounts of feeds that varied in fat level. And I'm going to really simplify this. I'm going to talk about two of the treatments One of those in blue was big fish with high fat feed. And this was the standard for this hatchery. And then small fish with low fat feed. And I really want to highlight here Lance Clark's role from ODF&W. He conceptualized the experiment, he initiated it, he carried it through. So basically this is Lance's study, and I just get the opportunity here to report on it. And so this is a visual. On the far left are the big high lipid fish. On the far right are the small low lipid fish. At release, the big fish weighed about 3 times more than the small fish. So there's a lot of difference between the smolt size. And those big fish are actually quite big. Those are big smolts, even for the Columbia River. Okay, this experiment occurred over 4 years, right during a big flip in ocean conditions. And in 2015, the last release year, the blob occurred. So the experiment was replicated over 4 years, but the years were not replicates. Okay, this plot is from the NOAA Ocean Salmon Indicator page. It's an average of ocean conditions over these 4 years. And basically it shows that 2012 was a very good ocean year, and 2015 was a very bad ocean year, and there are 2 intermediate years. So our experiment was really torqued by really different ocean conditions. You can see this by the salmon recoveries. This is just comparing recoveries across release years, all treatments combined. From 2012 to the 2015 release, there was a 10-fold difference in recoveries. And pointing you to the left axis here. So this is just recoveries per fish released. It's not survival. This is straight out of the database. Okay, we will look at the treatments, average over the 4 years. The first thing you should see is really big error bars. You can see that the big smolts did better than the small smolts, but that was not statistically significant. And in fact, the difference wasn't that big, or about 0.1%. So Lance carried a big load getting the experiment done. A lot of people said it was basically kind of stupid that the small smolts wouldn't do very well. And people have tried releasing small smolts before and it wouldn't work. But a big take home from this experiment actually is the small smolts did pretty well. They're roughly equivalent to the big ones. And that has some real implications for how many fish a hatchery can release, because you can rear more small fish than big fish. But these fish were also of different ages. 5 different year classes. You can see from the youngest to an age 4 fish here, age 5 and even age 6 fish are also recovered. They were actually too big to fit into the picture. So we're going to compare the treatments with the small low lipid and the big high lipid. The big fish recovered at an average of a younger age, about a half of a year. And I want to point out that I flipped the y-axis here. It's number of adults in an age class per number of adults rather than recoveries. Okay, so the big smolts come back younger, but on average, it's not that different. Okay, these young fish are really small, right? That age 2 fish is basically the size of a trout. The age 3 fish is maybe a coho. The age 4 fish is starting to look like a chinook. And the age 5 and age 6 fish are really pretty robust. So we just compare recoveries of age 2 and 3 fish between the treatments. The big smolts came back about a 5-fold greater rate of young fish. So the big smolts produce a lot of young males that are small. And the small fish, only about 5% of the adults came back as basically early maturing males. So this surprised me. This is a pretty big difference. If we compare the recoveries of age 5 and 6 fish, the small fish were recovered at a much higher rate in these large size classes, or about 50%. About 45% of the total fish recovered from the small treatment group were age 5 and 6 fish, and about 30% for the big high lipid fish. So the small smolts produce bigger adults. And that's what this shows graphically. The big smolts on the far left, the blue, came back as small males, whereas the small smolts in the orange on the far right came back older and larger. So again, this gets back to some of the assumptions of industrial smolt production. All smolts don't have the same performance. If you vary how you rear fish, what your fish look like, how big they are, when they're released, they will have different recovery characteristics. Okay, so overall for these results, there are huge differences between years. The average treatment recoveries were not different across years statistically, and this has some interesting implications for hatchery production. And there are really pretty interesting differences in the age of maturation and the age of recovery. So we go back to this thought experiment and thinking about how we limit variance rather than focusing on maximizing returns. Here we get to a point in the analysis that we're still working through in our group in Seattle, but it really intrigues me. This shows recoveries by year. So this is for the 2012 release. Each column over a year represents the fish recovered in that year. And the green are the large fat smolts, and the orange are these small skinny smolts. We can see that the big smolts are skewed to the left, and the small smolts are actually skewed to the right. You know, so that's what I've told you before. But what I want you to think about is the span of years over which the fish are recovered. And the way that the average age at recovery does not really reflect the variation we see in the age 5 and the age 6 fish and the increase in recovery span for the small smolts. So this is the same data display for the 2015 release. I'll toggle. So those are totally different, right? And not many fish came back from the 2015 release. We can still see that these small smolts had a greater span of years in which they recovered. So that was I think a decrease in variance as compared to the big smolts. So the take home here is I haven't quite figured out how to display these results in a way that I think makes good sense. Another take home is the years are really different. Third take home that's really important is it took 10 years to complete this experiment. And it'd be great to, do it again and see if the results are consistent. But 10 years is a long time, and that's not going to happen tomorrow. So really think about some modeling approaches to help us kind of focus these results and play with some scenarios. So again, thinking about what the hatchery goal is it maximizing returns or is it minimizing variance in returns? Maybe there's some things we can do to minimize the variance to kind of spread out the return of fish over years. Okay, so I'll finish up here. A quick summary is the oceans are very variable and it affects salmon production. We can introduce smolt variation that has implications for adult variation, both the numbers coming back and the size of the fish coming back. Some of the assumptions of industrial smolt production that hatchery programs were based on and still underlie a lot of hatchery production are not true. And in terms of thinking about hatcheries and where we want to go with them and what management goals are, really think about what the hatchery goals are. What do you want them to do first? And what strategies are used to meet the goals? So I don't have any clear answers. I don't have recipes, but I think we need to question what we're doing and why, and then think about how we're going to do it, if that makes sense. And so thank you much for your time, and I'll wrap up. I'm done.
So what we do is usually try and alternate questions to the two speakers, and I guess I can go first. And this question is for Brian Riddell. It comes from Gary Morishima. He says, the relationships and data presented are temporal. Do any of your analyses deal with identifying how environmental conditions affect survivals and any differences between hatchery and wild? And two, any insights into carrying capacity? There are some reports on the influence of pink salmon and other species.
Yeah, thanks, Gary. Yeah, some of the analyses that we've conducted tried to take into account area-specific environmental conditions. So we have sea surface temperature, salinity, local concentrations of predators. Another analysis is looking at the releases that are further upstream versus on the coast. And that what we find is that many of these don't really come out to be significant contributors to accounting for the variation. They can affect the difference in the average survival and that. So when we have included temporal effects basically in all of the models and that. Now, the carrying capacity is an interesting one and almost another talk, because what we're finding in the high seas is we don't see extensive overlaps of the different species. The really big mystery still is the absence of pink salmon. You use that example in your question. And in the workshops we've had with a number of the marine scientists across the North Pacific, I think there's an interesting consensus coming about that there's probably two major survival stanzas or periods, one being in the coastal zone where most of the salmon are moving northward. The other then is more in the high seas as they are growing over time for, growth and maturity. So the consensus, maybe a hypothesis, is that a lot of that competition may occur in the coastal zones in the early life phase. And we think it could actually be in the first year at sea, because what we're finding is that after the fish get into the first winter, we're definitely seeing stress in overwinter survival and their physiology. But in the absence of any real extensive overlap in the high seas at all, for competition to really occur, it's quite likely that it's mostly in the early life phase and in the very near shore. So this is a challenge that we've got in how we incorporate the salmon life history into some of the ocean and climate models now, because the coastal zones, of course, are very dynamic, and we probably don't have information at the spatial scales where some of these interactions are occurring. So it's a bit of a new challenge for us to think about. But we don't, I think a number of us safe to say, and Brian and Laurie have been in these conversations at times, I don't really think we're finding a lot of evidence that supports competition in the high seas. And that, so I'll leave it at that.
Thank you, Brian Riddell. So next, for a Gary question for Brian Beckman. Have you looked into attribution science to try to tease out hatchery from environmental effects? For example, the trends or changes in survivals and size also appear to be related to El Nino Southern Oscillation regime shifts.
Well, so the answer to that is really easy. No, I have not done that. At this point, it's still illustrating the variation and the range of variation and trying to put that together, and further analyses will follow.
Fair enough. Oh, okay.
Laurie,
Yeah.
just a quick comment on that, since we're interested in the environmental variation. One thing that has really impressed me over in the last 2 years of really talking with a lot of the climate modelers and how you relate that to the ocean is there are quite a few coastal zone climate models linking to the ocean processes and then to some of the fish species that I wouldn't call them predictive, but they are certainly explanatory. And so I think that the modeling groups have made significant progress. I think the biologists are the ones that are lagging them at this point in time. So some of the things that Gary is referring to, I think probably do have some opportunity to really investigate in the near future, probably within the next 3 to 5 years.
Good point. Brian Riddell, a question about how applicable your results are to other areas. And do you think the findings from southern BC would apply to the north, transboundary or Yukon rivers? Or do you think the data existed in the north would have different results?
Well, there's no question that we do see different results in different regions. We have not done the northern boundary. Well, no, I shouldn't say northern boundary under the PSC. We have not done the transboundary and the Yukon in any consideration yet. But with a very different life history for the Chinook up there, for example, you could see some differences. But I'm not sure that some of the things we're seeing in hatcheries are going to be easily avoided. I mean, the very high densities and the effects that Brian's talking about. I just think that given the environmental variation that we're talking about, we have to think of things differently. So on one hand, yes, there are differences in the big picture. I'm not sure if there aren't quite consistent messages amongst those areas that we have to be aware of. We'd honestly have to try, to be perfectly honest, because we really don't have much study up there for this sort of thing.
Thank you, Brian. So we have a rather long question from Steve Latham for Brian Beckman. So it's strange to me that there's resistance to science being done on hatchery fish. Is that because people think the value of what's learned won't make up for the loss of production? In the end, the results of the experiment show that the more expensive smolts to produce also generate the less preferred adults. We clearly don't know very much, and we ought to have experimentation as part of all of these hatchery programs. Brian Riddell indicated only a very small number of hatchery programs even had evaluation as part of their mandate. So would you like to comment on that?
Yeah, thanks, Marisa. And thanks, Steve, for the question. There is strong, I guess, fear of loss of production. People want adult hatchery salmon back for harvest. And there's, opposition to any loss of adult production. And I understand that. And, that's part of where society is. I strongly agree that experimentation should be a standard part of hatchery production at this point, especially given the variable ocean conditions. So I would advocate for variable production strategies within hatcheries consistently, because I don't think we'll find a best strategy going forward because things are changing and they're variable. So thank you.
Marcia, maybe I can add to that just briefly. The couple of diagrams I showed you from our first analysis that's now going to be published, you just look at the environmental noise that we're dealing with. And I showed you the long-term linear effect, but then you look at the extent of variation within a year across the various things. The whole concept of optimality, and that we can find optimums in our models, and they can increase some populations by a fair amount, others by very, very marginal increase at all. And that, so I think it's like Brian is saying, we really have to have more experimental, more adaptive type of approaches. And I think the bottom line to Steve's question is that it's just different values. The people that support these hatcheries, they don't want to acknowledge that there's uncertainties and that they may not be the best mean forward. There are different value systems we use, right? They might be fine for some aspects, but even in BC with the work that we've done with the genomics, we've had people say they don't want to try and apply them. They don't want to try selective breeding for returning to larger fish. I mean, the more effects that we show and the resistance to actually trying to improve it or change it, something's got to break. And that it doesn't make sense to keep doing the same thing that's not working.
Good point. I've had people tell me that my data is wrong because I was showing that 90% of the yearling Chinook coming out of the Columbia are hatchery fish. They said I made a mistake somewhere. So, and I follow up, Brian, a follow-up question from Steve for Brian Riddell. I thought it was interesting is the issue of mark selective fisheries and the fact that we can, selectively or select for marked hatchery fish, and whether in that way that the production, excess production can be accessed. And if, is that a solution to the hatchery problem or issue in dealing with this variable survival of given a variable ocean? And thoughts on that?
Well, my first thought is that we're mixing values there. Right. So if we're limited by conservation and the need to meet escapement goals for wild fish and have sustainable fishing, producing lots of marked fish at a time when the environment's limiting production is kind of counterintuitive. And that, not to mention the sort of effects on the assessment programs, but if you put a value on sustaining recreational fisheries for social and economic purposes, then marked selective fisheries probably have some role in some areas. And that, and again, I think it comes down to what your management objectives are or your social objectives. But there isn't any question that those marked fish can at some times be competing with the wild fish. And that, so I think you really have to be clear on what your objectives are when you enter into that game.
Thank you. So Brian Beckman, we have a question for you from the Yukon. So from the lipid experiment, are these Chinook ocean type, and how would the results be comparable in river type Chinook? So for example, in Whitehorse, they have mitigation hatcheries for a hydro dam, and they release large Chinook instead of staying a year in freshwater. This is done to minimize habitat competition. Survival has been poor, and if the hatchery switched to releasing smaller fry, could it possibly increase survival of the fry to adulthood?
Okay, thanks, Marisa. I'm rereading this. There's a lot to that question. They were ocean type Chinook. I think the results are definitely applicable to stream type or river type Chinook yearling releases. A fry release would be a different size and different age. And I'm not aware of any fry release hatchery programs that do quite well. The experiment was yearling fish of big size and small size released at the same time. So I don't think we can take that to releasing fry versus smolt. So it's kind of apples and oranges thing.
Here's one from Cheryl Lynch for Brian Riddell. I don't think one can talk about hatchery effectiveness and changes to population biometrics without also looking at possible impacts from hatchery harvest practices. Did the PSF team also look at changes over time to size and age in different harvest sectors and locations? So the kind of the influence of size selective harvest.
I think that's sort of a yes, no, Cheryl, because where we're looking at total returns, we do have to account for catch and escapement returns. So there could be a difference. There could be an effect there, but we have not looked at that in really close detail because particularly in the northern area where you get the very complex coastlines, you can have fisheries on the outside and mixed stock and that as opposed to more of the inside where you can have some really direct harvest on some populations, and that the harvest, the total catch, we've tried to account for the best we can, and these are all with agreed data sets and that. So I think the answer to that in all fairness would be depending on which populations you're looking at or species, and that there may be some that we can do that better for than others, to be honest.
Thanks. So this is a question for both of Brian, so perhaps Beckman can answer it first. It's from Scotty McPherson, and he says, how does the magnitude of hatchery production fit with the mandates in the PST to manage and sustain wild stocks, given that hatchery production around the Pacific Rim has led to the thought that there are more salmon in the Pacific than any known period?
Okay, I guess I got the first shot. So thanks, Scott, for asking an easy question. I appreciate that. A simple reply is that Chinook salmon fishermen in the Columbia River don't care how many sockeye salmon there are in Bristol Bay. So they're very strong regional desires for salmon that don't correlate to the whole Pacific Ocean. And as a NIMS employee, I'm very much aware of the conflicts between the mandates for wild fish conservation and harvest through hatchery production. And that's just hard. And I don't have any simple answers for that. And society really drives what we do as fisheries managers, and our decisions are, at the end made by politicians, no matter what we recommend. And that creates some strong conflicts.
This notion of that recently we've had the all-time high production of salmon in the North Pacific, the statement on the surface is true, but when you really look at the details, it comes down to a relatively small sample of species and areas. So it's largely what would be the Russian East Coast pink salmon and then Russian chum, and then Bristol Bay sockeye. The three of those account for probably at least 75% of the total numbers of fish. And in 2020, yeah, that's right. And in 2022, which most people won't have seen yet, you're going to see a massive drop. In 2020, it was a drop of 47% from recent year catches in total North Pacific, even though you had the Bering Sea was doing well for Bristol Bay sockeye, right? And then in 2020, it's going to be the same. And when you really look at the loss, it's Russian pinks. Used to have a huge contribution of Japanese chum from their hatchery programs, but their survivorships have really gone downhill to at least a drop of about 50% production. And what's really going on in the North Pacific is really with the hatchery and wild production in the east coast of Russia and Bristol Bay, and that as they mix in the North Pacific. And that, so it's an interesting comment, but it's not true widely. And you have others, Alaskan chum, Alaskan pinks, they can be very abundant at times. But the big changes that we're seeing in these year-to-year changes actually occur over in Russia, which is a real hit for us in trying to understand the North Pacific now with the Russian behavior in what am I trying to think of? The aggression they're showing. I doubt we're going to really have a collaborative program there for a number of years. So it's a significant concern for us in trying to understand the North Pacific.
Or do research out there with all the partners. Comment by Urs Thomas. He said he thanks both of you for great presentations, and for Brian Riddell, he's looking forward to seeing the future work on the impacts of spawning channels on wild populations like Babine sockeye. And I was just wondering, do you have an approximate date? You said it's not ready yet. Do you have an approximate date, or can you provide kind of a preview of some of the results so far?
Well, I think that the comments that we're going to make on the channels is going to be much less quantitative. There are some significant analyses going on in the Skeena, and they would involve Pinkett and Fulton channels in Babine Lake. So I think another group will be doing more of the quantitative analysis there. In southern BC, of course, we have a few channels in the Fraser and certainly down in Chehalis area. The channels are interesting in the sense that they're more natural and they can't produce a large number, but it's really a fishery impact there. You can't really utilize a lot of the production, and if you do, you're going to overfish some of the natural stocks. And I wouldn't say that that's necessarily still current, but there's no question over the last, it would be 20 to 30 years, that's been the principal effect of these channels. They can be very effective, but the fishing effect can be substantial on the local populations that are mixed with them. So interesting comment I received from Cam that this year with the drought we had in BC, a really good example, the big channels in Weaver Creek, which is in the Harrison River, the channel was not lined, and so the actual water table fell below the channel. So they couldn't actually even use the channel this year, although the facility was sitting there. Right. So these effects we're seeing with climate impacts are revealing all sorts of different impacts.
So to follow up on that question, Brian, a new question from Don Hall regarding the effectiveness of hatchery production for rebuilding stocks. I think that he says, I think I heard, and he's correct, it is not effective in rebuilding wild populations. Currently a major program in BC, the rebuilding aspect. So what are alternatives? And he also says, thanks to both speakers, look forward to the final results and report.
Oh, I think, Don, you should be retired by now anyhow, but you did grab the key point. To me, that's one of the great disappointments. We've looked at a large number of rivers now where there is mixture with hatchery fish going in, both direct and indirectly, and that. And once the input of hatchery fish stops, you see an immediate drop in the number of fish spawning naturally, and then in the next generation production. So if you can't rebuild through hatchery production, then the second part of his question is very appropriate. What do we have to do? I think it's coming down to referring to the habitat, the water content to start with, stability of the channel sides, and that how we restore the stability of the riparian zones, for example. But bottom line is you got to get fish in there to spawn, and you've got to give them that opportunity. But the major hatcheries across the board, we're not seeing any, well, I can't say any, but we're seeing very few examples. Now, some of the, it was interesting at a previous discussion on this, is one of the things that is pointed out is you do get them through a bottleneck. So we don't know the what you call the parentage of the fish that are there. You could do that now with some of the genetic methods. And if you really wanted to look at this, it's quite possible that some of the fish from the hatchery do have ongoing progeny in those systems, and that the system would have been poor for it if you didn't have those spawners a couple of generations back. But we're certainly not seeing evidence of active rebuilding.
Brian Beckman, do you have any comments on supplementation for rebuilding wild populations?
I'm thinking about comments I would like to offer to a public venue. Certainly that's a driver for many salmon hatcheries down the Columbia River. And yeah, I look forward to seeing how effective they've been. How's that?
Non-committal.
That works. Well, we've come to the end of our question and answer session. I just want to thank both speakers. Those were fantastic talks, really lots and lots of food for thought. And our next seminar, we're not going to do a December seminar because of the holiday, but our next one is actually going to be during the meeting in Vancouver on Tuesday, January 10th. And we're going to be talking about the Salish Sea Survival Project is the theme. And we'll have Isabel Pearsall with the Pacific Salmon Foundation and Katie Bizyayevi with the Stillaguamish tribe will both be talking. So it'll be an in-person event finally. And then we're also going to have in-person seminar in February talking about the results of the CSC's survey of use of environmental indicators. So really excited to be able to all be in the same room at the same time again. And with that, we'll sign out, and thank you again to the Bryans for a really excellent seminar series.